So exam in here tomorrow. Do as before, the every other seat, starting with those at the end as the odd number, and then move in. Stay on the odd numbers. Somebody told me a joke earlier today. How did that go? Start out with a joke. And maybe that's what you guys need today, right? How did that go? I don't even remember it. Oh, man, this is scary when I start forgetting jokes. I'll tell you one I made up. Okay, you guys ready for one I made up? All right. So it's about this meteor hunter who was looking for where the meteor landed. And when he finally found it, it was a groundbreaking discovery. You're laughing. Okay. Well, hopefully you're not laughing at me. You're laughing with me. But in any event, I'll take laughter whatever I can get, I guess, right? All right. So yesterday I finished talking about gluconeogenesis, and I decided that that was going to be the end of the material for the exam. So where I'm starting here will be what we call the home stretch, which is the last material for the final exam. The final exam is comprehensive, and the final exam, I will give you a note card that you can fill out with anything that you want on that note card, so long as you do it all in your own handwriting. You can't print things with a printer on a note card, for example. Okay? You will have to get the note card from me. I'll bring it to class, but you will have to get the note card from me. And if you come to the final exam without a note card that you got from me, you will lose points. So if for some reason you're not in class, which I know nobody ever misses class, but if you ever miss class and I brought the note cards, then that will mean that you'll need to come get one from me in my office. Because you will lose points if you come to the final without a note card you get from me. Does that make sense? Even if you put nothing on it. All right? So the requirement is coming to the final exam with a note card you got from me. Whatever you want to put on it is fine, as long as you do it in your own hand. Okay? I'll bring those sometime soon. I won't tell you. That way you come to class, right? You guys are good about coming to class. All right. What I started to tell you at the end of yesterday was a cycle that goes on in your body that students understand really well after I explain it to them, hopefully. And it's really kind of a cool cycle that tells you how your body is set up to function and give you things that you need. All right? Let's think about, for a moment, your liver. Right? Your liver is doing incredible things. And your liver is helping to control the level of glucose that your bloodstream has. Your liver is taking up glucose when there's too much glucose. Your liver is releasing glucose when the body needs glucose. Your body is handling fats as the body needs energy, releasing them as needed, taking them up when you eat them. Your liver is modulating your cholesterol, which is really important because your cholesterol is necessary for making membranes, and getting the right level is very important. And we'll talk a little bit about that in just a bit. Okay? So your liver is a pretty remarkable organ. Very, very remarkable organ. And the liver plays a role in this cycle we call the Cori Cycle. Okay? So the Cori Cycle was a very interesting cycle. It was discovered by a husband and wife from, I believe, Austria or Germany. I can't remember where they were from. There was OSU Theater did a play about them about a year ago. Does anybody see the Cori play? Does anybody go to OSU Theater? No. OSU Theater is pretty cool. They've got some good stuff. Anyway, so they did a play about this husband and wife couple back in the late 40s, early 50s, who discovered this cycle. And they were an unusual team, especially at that time, because women were not working in laboratories much. And so they were pretty cool. Anyway, so here's their cycle, all right? So it tells us that within the body, okay, you got it?
No. I think it's up to you.
Okay. In the body, gluconeogenesis runs in some places, and in other places, glycolysis runs. And they run to your advantage. So let me describe how that works. Let's imagine that you decide to go out for a run in the morning like I like to do. And you start exercising, and you start running faster than your body can supply oxygen to your muscles. You can do that reasonably well. Okay? you can run faster than your body will deliver oxygen to muscles. Now, one thing I didn't tell you yesterday, which means you don't need to know it for the exam, but you will need to know it for the final, is that when you burn glucose in the absence of oxygen, it's extremely inefficient. I'll give you an idea. We're going to see what happens with pyruvate. Pyruvate is the end product of glycolysis. If we have oxygen, pyruvate will go to the citric acid cycle and be oxidized ultimately to carbon dioxide. And that process in the citric acid cycle generates a lot of ATP. Not directly, but indirectly. A lot of ATP. You saw that if we went through fermentation, pyruvate got converted into lactate, and we produced, the only ATPs that were produced were those that were produced during the cycle of glycolysis, and that was 2. Right? Well, it turns out that if you are going through fermentation, for each glucose, you will get a grand total of 2 ATPs net. And if you take that pyruvate and you run it through the citric acid cycle, and by the way, you can't do that unless you have oxygen. So when you run out of oxygen, you can't run pyruvate through the citric acid cycle. Okay? If you run pyruvate through the citric acid cycle, and you add the ATPs you get there to the 2 ATPs that you got in glycolysis, you end up with 38 ATPs. And people vary in how they count that. But you essentially have 38 ATPs that are produced by the complete oxidation of glucose in the presence of oxygen compared to 2 ATPs in the absence of oxygen. You're getting 19 times as much ATP when you have oxygen as when you don't. My question to you is, if you want to lose weight, do you want to go aerobic or anaerobic? I hear 1 vote for aerobic. Do I have any votes for anaerobic? 1 vote for anaerobic. Okay, that means we have to vote on as a class. So if you want to lose weight, do you want to go aerobic? Or do you want to go anaerobic? Okay? You want to go anaerobic, folks. And the reason you want to go anaerobic, you want to lose weight, right? You want to generate ATP in the least efficient way because you're going to burn the most stuff up, right? So you want to go anaerobic. We have this fascination with aerobic. When most people say they're going aerobic with their exercise, what they're really doing is going anaerobic. That's why they're breathing heavily, because their body is trying to supply the oxygen that's not being supplied. Okay? They think they're going aerobic, but they're really going anaerobic, and that's why exercise helps you to keep the weight down. That's cool, right? All right, well, this brings me to this slide, the Cori cycle. Okay? What do muscles need for exercise, for contracting? They need ATP, and where do they get ATP? They get ATP from glucose. The maximum amount of ATP that they're going to get is when the muscles are completely supplied with oxygen, and the least amount of ATP that they get is when the muscles are not adequately supplied with oxygen, right? You've heard of carbo-loading? Carbo-loading starts to make some sense. Carbo-loading is what long-distance runners will do before a race. They will eat a lot of carbohydrates because they're trying to load their muscles with glycogen. And glycogen is a storage form of glucose that can be released as needed, right? All right, so I'm out running. And if I'm really good, I'm exercising faster than my blood supply can deliver oxygen to my muscles. My muscles start going anaerobic, all right? When my muscles start going anaerobic, they're going to start burning 19 times as much glucose as they would if it were aerobic. My muscles need ATP. My muscles need a lot of glucose. My muscles have glycogen, so they can start breaking that down, but it sure would be nice if they had an external source of glucose, and they do. Okay? This is where the Cori cycle comes in. All right? Let's forget the muscles for a moment. Let's think about what's happening in the liver. Your liver is close to your lungs, right? And your liver is fairly well supplied with oxygen. Your liver has plenty of oxygen compared to your muscles when you're exercising. Your liver also has a lot of glycogen. Okay? And your liver is capable of releasing glucose. Okay? But it turns out the liver doesn't really have to do that. It doesn't have to release glucose from its glycogen. Why not? Well, what are muscles doing? Muscles are exercising. They're running out of oxygen, and what's the byproduct of muscles from glucose when they run out of oxygen? What do they make? Lactate, right? Muscles have zero use for lactate because it's a dead end. The only thing you can do with lactate is convert it back to pyruvate. But that takes oxygen, and muscles don't have it. So what do muscles do with lactate? They let go of it. They release it into the bloodstream. And the lactate travels in the bloodstream, and it goes and it hits the liver. And the liver says, oh, I've got plenty of oxygen. Let's convert this lactate back into pyruvate. And since we've got plenty of oxygen and since we've got plenty of energy, let's take that pyruvate and convert it back into glucose, gluconeogenesis. So the liver is taking the lactate that the muscles are releasing, converting it into pyruvate, and then converting it into glucose, and releasing the glucose, which goes back out to the muscles. I've just described the Cori cycle. Okay? It works, and you might say, well, how do you have an anabolic pathway and a catabolic pathway going on at the same time? I thought you said that was bad, Kevin. It is if it's in the same cell. It's not going on in the same cell. It's going on in different parts of the body. Liver's doing gluconeogenesis. Muscles are doing glycolysis. Make sense? Questions? Yes? Say it again. Yep. So do you get lactic acid buildup in your muscles because the liver can't process it? That's probably not the case. It just means that the muscles are releasing a lot of lactic acid. Some people think that lactic acid is what causes muscles to be sore after you exercise heavily. There are some people who argue with that, but that's one thought about why you're sore after heavy exercise. Yep. Okay. So that's the Cori cycle. Very cool. Okay. I lied to you a little bit yesterday. I told you a little bit about how pyruvate kinase was regulated, but I didn't tell you the whole story. Pyruvate kinase is regulated in an interesting fashion. It's regulated allosterically, meaning it binds to small molecules like fructose 1,6-bisphosphate, like ATP. I didn't tell you about alanine, but alanine also will affect the enzyme, and you can see it's affecting the enzyme negatively. Okay? But it can also be regulated by covalent modification. Phosphorylation of pyruvate kinase causes it to be less active. Dephosphorylation causes it to be more active. We'll talk later about enzymes that phosphorylate other enzymes in just a little bit. Okay? Why do you suppose that pyruvate kinase is regulated at all? It's the last step in the pathway. Does that make any sense? It does? Good. It does help control the direction. You're right. But there's a more important reason why this enzyme is regulated. It's really important for turning off glycolysis. Do you know why that would be important? You're on the right track. Okay. So it's important to turn off glycolysis when gluconeogenesis is going on. And you say, well, can't PFK do that? Doesn't PFK do that? It does. PFK will stop glycolysis, but it will allow gluconeogenesis to occur. But let's think what's happening in gluconeogenesis. In gluconeogenesis, we're starting with pyruvate. We're going to oxaloacetate. And then we're going to PEP. And what happens to PEP in glycolysis? That's the Big Bang, right? What we're doing here is we're turning off the Big Bang. Because if we don't turn off the Big Bang, what's going to happen is we're going to make PEP, Big Bang. Make PEP, Big Bang. we've now made a little miniature feudal cycle. Make sense? So regulating pyruvate kinase allows the cell to turn off that last reaction that's the Big Bang reaction so that gluconeogenesis can proceed without getting interrupted. Everybody understand that? Okay, nobody's going to say anything today because they're thinking about tomorrow. Okay, let's see. Let's talk about another pathway, and I'll just throw this up here to scare your pants off of you, okay? This pathway is called the pentose phosphate pathway, and we're not going to go through it step by step. But I'm going to tell you some of the things about the pathway that's kind of interesting, okay? It's called the pentose phosphate fat pathway. First of all, what's a pentose? 5-carbon sugar, right? Okay? It's called the pentose phosphate pathway because if you look through it, you'll see several 5-carbon sugars that are being made and broken down. Did you tell a joke? Oh, okay. Did you tell a joke? No. The pentose phosphate pathway is making pentoses, and one of the most important pentoses in our body that the pentose phosphate pathway has to make is ribose. Because we need ribose to make nucleotides. The pentose phosphate pathway is the cell's way of making ribose. In fact, it makes it as ribose 5-phosphate that you can see right there. Ribose 5-phosphate. The reason I don't spend a lot of time talking about the pentose phosphate pathway in terms of reaction to reaction is that this is a pathway that doesn't have one specific direction. It can go in many different directions depending upon the needs of the cell. We think of glycolysis. We have a starting point of glucose. We have an ending point of pyruvate. We'll see later that the citric acid cycle is a circle. We start with oxaloacetate, and we end up with oxaloacetate, and it goes around and around and around. Okay? But this pathway has numerous starting points, and it has numerous ending points. Let's start at one of the starting points. Here's a starting point, glucose 6-phosphate. Remember I told you yesterday that glucose 6-phosphate was used in some other metabolic pathways? Here's one of them. Glucose 6-phosphate can be oxidized to 6-phosphogluconate. No, I don't expect you're going to memorize that. Okay? I expect you should know glucose 6-phosphate's in this pathway. But I tell you about this oxidation because this oxidation uses NADP and produces NADPH. A second function of the pentose phosphate pathway is it's one of the most important sources in the cell of NADPH. We'll see that NADPH is very important for making fatty acids. So NADPH has to be made somewhere. This is one of the most important places that cells make NADPH. Okay? I've told you about ribose, right? I've told you about NADPH. And you can see that there's a bunch of other sugars that are up here. You can see some glycolysis intermediates. Here's glyceraldehyde 3-phosphate. Here's fructose 6-phosphate. Here's glyceraldehyde 3-phosphate again. There's erythrose 4-phosphate. You see various sugars of various sizes that the cell can mix and match and use as it needs. So the question is, which direction does this pathway go? The answer to the question is the pathway goes the direction that gives the cell what it needs the most of. If the cell is needing NADPH, this reaction is going to go like crazy. If the cell is needing ribose, it's going to be making this, and it could start with any of these things and end up at ribose. It's a very flexible pathway that allows sugars to be interconverted and, most importantly, converted to ribose 5-phosphate if the cell needs it. Okay? So the pentose phosphate pathway, very important in this respect. We'll talk later about the Calvin cycle, which is a pathway of photosynthesis known as the dark reactions of photosynthesis. And you'll see many of the intermediates that you see here in the Calvin cycle as well. Okay, so much for the pentose phosphate pathway. Okay, let's turn our attention to glycogen. I've alluded to it already once, and I'm now going to tell you more about glycogen metabolism in some detail. Glycogen is, of course, the polymer form of glucose that animals use to store glucose. We store glycogen mostly in our liver and our muscles. That's the most common places we find glycogen. Okay? Glycogen is important because glucose is a poison, but it ceases to be a poison when it is in the polymeric form of glycogen. I'll remind you that it's a polymer of glucose with alpha-1,4 bonds and some alpha-1,6 branches. About every 10 glucoses, we see a branch. So it's a very branched molecule, and this figure is attempting to show you the branching of glycogen. Okay? Glycogen metabolism pathways is the easiest pathway you'll ever learn. Okay? Everybody smiles when I say that until I start telling them about the regulation. But the pathway is really simple. Here's glycogen. Okay? It's a polymer of glucose. You see that little N right there, meaning we got many, many, many glucoses there. Glycogen is broken down by an enzyme called glycogen phosphorylase. You saw it earlier in the class. I'm going to tell you more about it right now. Glycogen phosphorylase does something really interesting. Okay? It takes a phosphate, and it uses that phosphate to clip off a glucose. It's a reaction we call phosphorylasis. P-H-O-S, P-H-O-R-Y-L, O-L-Y-S-I-S. I'll spell that again. P-H-O-S, P-H-O-R-O-L-Y-S-I-S. Phosphorylasis. Okay? Phosphorylasis uses a phosphate to break something. In this case, the breaking is it's breaking a single glucose off of glycogen, and in the process, that phosphate gets attached to glucose at position 1. We make something called glucose 1-phosphate. Okay? There's an enzyme called phosphoglucomutase, and yes, you should know the name of that enzyme as well. Phosphoglucomutase that will convert glucose 1-phosphate into glucose 6-phosphate. Yes. Phosphoglucomutase converts glucose 1-phosphate into glucose 6-phosphate. Why does the cell want to do that, do you suppose? What's glucose 6-phosphate useful for? Go ahead. Glycolysis. Because remember, the cell is breaking down glycogen because it wants to either burn glucose to provide it for the muscles that need it, or in the case of the liver, to release glucose so that the blood glucose levels can be increased. Okay? We've just completed the breakdown of glycogen, well, with the exception of one thing. Okay? The one thing is that glycogen phosphorylase is an odd enzyme. Glycogen phosphorylase is really good at breaking the ends of glucose off, but it's not very good at working very close to a branch. Here's a branch. There's the polymer, alpha-1,4 linkages. There's a branch, alpha-1,6. It turns out that glycogen phosphorylase works really well until it gets within about 4 or 5 residues of a branch. It stops at that point. It won't go any further. If the cell didn't have a way of resolving that branch, every time that the phosphorylase came to a branch, everything would stop. Okay? Well, it turns out that the cell has another enzyme to deal with branches. It's got a name that's about this long. We're going to call it debranching enzyme. From the name, I'm hoping you could guess what it does. What it does is what you see on the screen. It takes part of the branch, shown in blue, and it moves that part of the branch down here. So it's taking it from the top strand to the bottom strand. It's taking a 1,4 linkage here and making a 1,4 linkage here. And you'll notice what's left behind. A single molecule of glucose in a 1,6 linkage. Okay? The same enzyme will clip that guy off right there and give a free molecule of glucose. This is the only place in glycolysis where free glucose is released. Okay? Now, the phosphorylase says, oh, look, I can cut out some more, cut out some more, cut out some more, cut out some more. And so the breakdown of glycogen proceeds in the way that I just described to you. Phosphorylase works until it gets to a branch. Branching enzyme basically breaks the branches. And then phosphorylase comes back in and moves again until it gets close to another branch. That's all there is to glycogen breakdown. 3 enzymes. Glycogen phosphorylase, phosphoglucomutase, and debranching enzyme. Okay? At this point, we have glucose 6-phosphate, and we have glycogen that's been shortened. Okay. You want to make glycogen? It's just about as simple. There's only one more enzyme. Or one larger number of enzymes. There's other enzymes. But making glycogen is a function of an enzyme called glycogen synthase. S-Y-N-T-H-A-S-E. Glycogen synthase doesn't start with glucose. And it doesn't start with glucose 1-phosphate. In fact, to make that polymer bond, that alpha 1,4 bond for the glucoses in glycogen, to make that bond requires energy. And the energy comes indirectly from a molecule called UDP-glucose. I'm going to show you what the molecule is, then I'll tell you how it's made in a minute. Okay? This molecule is a substrate for glycogen synthase. Meaning, glycogen synthase is using this molecule to add a glucose, which is right here on the end, to a growing glycogen chain. And the energy comes from the fact that this is a nucleotide, that's the UDP part, with a couple of phosphates on it. This is a high-energy molecule. When glycogen synthase takes the glucose off of here and adds it to that growing glycogen chain, energy is released, and that energy is used to add the glucose to the growing glycogen chain. This molecule is an example of what I like to call an activated intermediate. An activated intermediate. An activated intermediate we'll see in other places in biochemistry is a molecule that has a high energy bond in it. And it uses the energy of that bond to donate a part of itself to something else. I'll repeat that. An activated intermediate is a molecule with a high energy bond. And it uses the energy of that bond to donate a part of itself to something else. In this case, the something else is glycogen. And the high energy bond is this bond between the glucose and that phosphate right there. Question about that? Okay. Okay. Well, how does this molecule get made in the first place? Okay. This molecule, actually I don't have it on here. This molecule gets made by another enzyme. Okay. The enzyme has a mouthful name as well. The enzyme is called UDP-glucose pyrophosphorylase. I will give you naming rights to it if you would like. Kevin has been taken. What shall we call the enzyme that makes this molecule? I'll tell you what the substrates are in a minute. Brian? Ryan. We have one vote for Ryan. My wife's name is Indira. We'll call her Indira. Okay. There's a vote for Indira. And what? Bob. Bob the enzyme. Bob, Ryan, Indira. Should we vote? Okay. Votes for Bob. Bob looks pretty good. Votes for Indira. I think Bob's beaten Indira so far. And votes for Ryan. Okay. I think it's Bob. All right. So you can call this guy Bob. All right. Not this guy, the enzyme that makes this guy. So what does it take to make this guy? Okay. To make UDP-G, which is what abbreviation is you can use here, to make a UDP-G, you need two molecules. You need a glucose 1-phosphate, and you need UTP. UTP is a ribonucleotide. Yes, the two molecules are glucose 1-phosphate and UTP. So Bob catalyzes the formation of this from those two molecules. All right. So this is the substrate then for glycogen synthase. Glycogen synthase is going to take this molecule and add the glucose to a growing glycogen chain. Okay? Now, here's the reaction. Here is UDP-glucose. Here is a glycogen chain. And here is that glucose being added to that chain, and you see that it's gone from this down to this. The glucose chain has grown, the glycogen chain has grown by one glucose residue. Notice that it made an alpha 1,4 bond. We're almost done with glycogen synthesis. What's the one thing that we need? Branches. We need to have branches. And you're going to like this. The enzyme that makes branches, we're going to call branching enzyme. See, this is all simple. Branching enzyme. You now know all the enzymes necessary to make and break down glycogen. That's pretty cool. Pretty simple. Doesn't take anything more than that. Making and breaking down glycogen happens with all those enzymes. Okay. Here's branching enzyme. What does branching enzyme do? Well, if you look at the various books, they'll tell you exactly how many sugars in it is and where it makes the attachment and so how far apart they have to be. And the reality is, for our purposes, it doesn't matter. We know that it makes branches on the order of about every 10 glucoses. This is showing a little closer, and that's fine too. Okay. And at this point, you've got glycogen. Okay. Now, I alluded to the fact that the regulation of this pathway is somewhat more complicated. Okay? You guys need a joke? Yes. I know if I ask that question, the one way I will get you guys to say yes. Will you give me your bank account? Can I make a joke? Yes. Right? Just make sure I ask those two very quickly. All right. I'm running out of jokes, but here's my joke for you. This is the joke about the two hunters that go out and get lost. All right? So there's these two hunters, and they come up to this farmer's house, and they say, "Can we hunt on your land?" And the farmer says, "Yeah," but he says, "I'll tell you what." He says, "A lot of people go out there. It's really heavily wooded. You go out there, you're going to get lost." Hunters say, "No, we don't. We hunt all the time. We don't get lost. We're not going to get lost." And the farmer says, "You know, most people get lost. Most people say exactly what you're saying, and they go out and they get lost. No, it's not going to happen." They argue anyway. And the farmer says, "Okay, I tell you what. You go out there, you get lost, which is going to happen. You shoot 3 times in the air. I'll find out where you are. I'll come out and get you, okay?" And the hunters say, "Okay, this is a pretty good deal." So they go out there. They go hunting, and it's starting to get late in the day, and they discover that they are lost. You saw this is coming, right? They discover that they're lost. And so they're kind of embarrassed to admit the fact that they're lost. And so one of them finally says, "Hey, man, if we don't get out of this place, it's going to be dark, and we don't want to be stuck out here after dark. Shoot 3 times in the air." So the other guy shoots 3 times in the air, and they wait, and no farmer shows up. It's getting closer to getting dark. Shoot 3 more times in the air. Shoots 3 times in the air. No farmer. No nothing. By this point, they're getting really unhappy with the farmer. Right? It's starting to get, twilight, so they're really getting close to getting dark. The other guy gets worried, and he says, "Shoot 3 more times in the air." And the guy says, "I will," but he says, "I tell you what, I'm almost out of arrows." A little laughter anyway. Okay. Let's talk about this, then I've got a song for you, and then we'll go home. How about that? Okay? This pathway explains what happens or how glycogen breakdown is controlled. Glycogen breakdown in the cell is something that's a very big commitment for a cell. Why? Well, glycogen is loaded with glucose, and glucose is a poison. So the cell wants to be careful to only let out as much glycogen as the body needs, and to let it out and be able to stop the release of that glucose as quickly as it can. So glycogen phosphorylase turns out to have 2 very big controls on it. We saw this with pyruvate kinase, only now I'm going to tell you about how this enzyme works in more detail. Okay? In pyruvate kinase, I told you that the enzyme was both allosterically regulated and regulated by covalent modification. Glycogen phosphorylase is regulated allosterically and by covalent modification. Let's talk about the covalent modification first. The covalent modification is pretty straightforward. Either we add phosphates or we take away phosphates. Okay? We either add phosphates or we take away phosphates. The form of glycogen phosphorylase that has no phosphates is called phosphorylase B. Most people refer to the phosphorylase B form as the less active form. We'll see why in a minute. Most people refer to the phosphorylase A form as the more active form. And the A is, of course, the form that has the phosphates. Well, first of all, how do the phosphates get on, and how do the phosphates get off? The phosphates get on by action of an enzyme called, it's not even on there, phosphorylase kinase. Okay? Phosphorylase kinase is activated by a pathway that's connected to epinephrine, adrenaline. When you get scared, your body makes adrenaline. Adrenaline is a hormone. It travels in your blood. It goes to the target tissues, and it stimulates phosphorylase kinase. And phosphorylase kinase converts glycogen phosphorylase B into glycogen phosphorylase A. Notice the phosphorylase is the enzyme, not the glycogen. Right? We're converting phosphorylase B into phosphorylase A. This has the effect of creating a more active form of glycogen phosphorylase. Release of adrenaline is therefore stimulating the breakdown of glycogen. Blood glucose levels rise. When blood glucose levels rise, muscles have more glucose to do what they need to escape danger, and everybody is well served. Let's imagine that you eat a big meal, and you eat a big meal, and you've had a Big Mac, and you've had a ton of fries, and you're having that giant mega-sized Coca-Cola that goes with that. What's happening to your glucose levels in your body? Glucose levels are going up, and glucose is a poison, so your body says, I don't like that poison. The body says the body's response to increasing glucose levels are to release insulin. The pancreas releases insulin, and when it releases insulin, cells are stimulated to take up glucose, so blood glucose levels fall, but glucose is a poison. Cells have to do something with that glucose they take up, and insulin also stimulates the dephosphorylation of glycogen phosphorylase by activating phosphoprotein phosphatase. This enzyme in the bottom is stimulated by insulin. The enzyme on the top is stimulated by adrenaline. Yes? Recap all that. Okay. Yeah, that's fine. Okay. You may start with the eating a big meal? Sure. Okay. Eat a big meal. Blood glucose levels, boing. When blood glucose levels go up, the body's response to that is got to get rid of this glucose. The pancreas releases insulin. Insulin tells cells, take up glucose. Basically swallow this poison so that our blood supply of glucose is not too high, because we don't want to damage our organs. That's what's going to happen if we have too much glucose floating around in the blood. The cells take up the glucose, but they've got to do something with it, and what they do with it is they make glycogen. Okay? If they're making glycogen, they don't want to break down glycogen, so they pull the phosphates off of phosphorylase A to make phosphorylase B. This enzyme, phosphoprotein phosphatase, is stimulated by insulin. We go from right to left with insulin. We go from left to right with epinephrine, adrenaline. So we're talking about putting phosphates on, adrenaline, taking phosphates off, insulin. Okay? Now those are stimulating that happening. Insulin doesn't physically take the phosphate off, and adrenaline doesn't physically put the phosphate on, but it stimulates the enzyme and the enzyme. Insulin on the bottom, epinephrine on the top. Okay, well, what about the up and down? Up and down, we're going from T form to R form, or R form to T form. Right? Let's focus on this one on the left, the dephosphorylated form of glycogen. We've got phosphorylase B. This tells us that some things will cause the phosphorylase B to be in the R form, and some things will cause it to be in the T form. The T form is the least active form, correct? I've got the least active form, and I've got the least active form. This guy up here is not going to be very active. What does it cause phosphorylase B to go into the T form? ATP or glucose 6-phosphate. What's ATP an indicator of? High energy. Do we want to be breaking down glycogen when we have lots of energy? No. We turn off glycogen breakdown. When my muscles get exercising and they need energy, their AMP levels rise, and AMP is an indicator of low energy. And what does AMP do? It converts phosphorylase B from the T form, very inactive, to the more active R form. This guy is active. This will break down glycogen. Very cool. Well, how about over here? How about this guy? The glycogen phosphorylase A can be converted from the R form into the T form with glucose. What's a glucose an indicator of? It's a trick question. It's an indicator of glucose. Glucose is a poison. Do you want to be breaking down glycogen when you've got plenty of poison? You convert glycogen phosphorylase A into the less active form, the T form. Okay? Well, how about going down? You'll notice there's nothing going down. This tells us that if there's no glucose present, that the T form will automatically convert into the R form for phosphorylase A. This is why we think of the A form as being more active, because usually the cell doesn't have much glucose floating around. Usually when we have glycogen phosphorylase A, it's in the R form. Now, why does the body have both of these systems of control? This is what I'm going to finish with. Okay? Why does it have both of these systems of control? Both covalent modification and allosteric regulation. The answer is because it serves the needs of the body best, and I'll tell you why. I told you left and right was either adrenaline going to the right or insulin going to the left, right? These are hormones. Hormones travel in the body, in the bloodstream, and they take on the order of seconds to go from the place where they're made to the target tissue. Seconds. That's why when you see the person who sees the baby under the car and they go to pick up the car, they don't do it instantaneously. They have to have their blood glucose levels rise before they can have that happen. It takes seconds. Hormonal processes, left and right, okay, take at least seconds to occur. Need, however, may be instantaneous. If you see somebody chasing you and coming after you and you've got to go right now, you don't want to wait for hormones to kick in before you can get away. Instantaneous needs are the up and down. So your body has both of these systems built into it because they help you at the best at the times that you need them. The hormones can deliver a lot of glucose, as we see with the picking up the car. The allosteric interactions don't activate as much. So the hormones are slower, but they really give a lot of stuff. Questions about that? Okay, I have a song about this, and I'm not going to sing it. I've got somebody else who sings it better than me. And let me turn on that. There's that buzzing noise. And here we go. It's called In My Liver. All right.